Hi and Assalamualaikum. My name is Hanif Nazmi bin Shamsul Zaman. I am from class AT11001A. Today I am going to talk about I want to present about monocotyledon and dicotyledon, or we known as monocot and dicot. Okay, so let's start with monocotyledon. Okay, what is monocotyledon? Monocotyledon, or monocot for short, refers to one of two groups of flowering plant, or we known as angiosperms. Okay, so as you guys know, most flowering plants are traditionally divided into two different categories, which is monocot and dicot. Members of each group tend to share similar features. Monocot, as the name implies, are defined by having seeds that contain a single monoembryonic leaf known as cotyledon. This is a monophyletic group that constitute a majority of our agriculture biomass and include many important crops, staples, but not limited to rice, wheat, corn, sugar, and others. A word of caution: when classifying flowers into monocot and dicot or dicots, remember that there are always exceptions to the rule. Some monocot may have a feature typically found in dicot or vice versa. There's a sixth way to distinguish monocot from dicot. Flower, leaf, root, stem, pollen grain, and the last one is embryo. Okay, so the first one is flower. Flower usually arrange their parts in circle with the reproductive parts in the middle, surrounded by petals and sepals. In monocot, these flower parts are trimerous. In other words, the flower part of a monocot are arranged, structured, or numbered in multiples of 3, usually with one stigma, 3 stamens, 3 petals, and calyx formed by the sepals in number less than or equal to the number of petals. Okay, the second one is leaf veination. Veination refers to the pattern of the vein in a leaf blade. These veins are responsible for the transport of water and carbohydrates throughout the plants. In monocots, these veins are arranged in a like parallel pattern. So, parallel veins are smaller in size with even smaller veins connecting them. Number 3 is roots. While monocot starts off with a taproot, this taproot tends to die soon after germination and replaced with adventitious roots. Adventitious roots look fibrous and are spread widely throughout the soil in many different directions. They tend to occupy the upper layer of soil and can be modified for different purposes like additional anchorage or aerial support. Because adventitious roots typically arise from an organ that is not the root of a plant, such as the stem, or sometimes a leaf, we are able to grow multiple plants from stem or leaf cutting of a pre-existing plant. Okay, so number 4 is stem. Okay, it is important to note that the stem of monocot have lost the ability to increase their diameter by producing wood and bark through secondary growth. Instead, monocot stems die down each other year, allowing new stems to grow. The only growing point of a monocot stem is at the top of the stem, disallowing the growth of any side stem or branch. Typically, then, monocotyledons are small and herbaceous. In a cross-section of monocot stem, you will find an epidermis, hypodermis, ground tissue, and vascular bundles. Monocot stem have the following characteristic, that is single layer epidermis with a thick cuticle, lack of epidermal hair, lack of concentric arrangement. Hypodermis is sclerosematous, presence of bundle sheets, oval vascular bundle of different size, and most notably scattered vascular bundle that do not have create any pattern. So number 5 is pollen grains. Monocots have pollen structure that is retained from the first angiosperm. The pollen grain of monocot is monosulcate, meaning that the pollen has a single furrow or pore throughout the outer layer. Okay, last is number 6, embryos. Embryos, part of the seeds that contain all of the precursor tissue of the plant and one or more cotyledon. As the name suggests, monocots are categorized by having one monocotyledon in the seed and one leaf emerging from the cotyledon. The seed pod of a monocot is also trimerous, or we know as in part of tree, because the carpel from which they grew also consisted of 3 parts. The cotyledon is the first part of the plant to emerge from the seed and is the actual basis for distinguishing the 2 main groups of angiosperm. Cotyledons are important in food absorption and are responsible for absorbing nutrients from the environment until the plant can photosynthesize its own nutrients. Okay, so let's continue with dicotyledon. What is dicotyledon? Dicotyledon, or dicot for short, refer to one of 2 main groups into which flowering plants, or we known as angiosperms, are categorized. Dicot, as the name implies, are named number of cotyledon or embryonic leaf that found in the seed. Embryo, they have 2 or D cotyledons. Unlike monocots, dicots are not a monophyletic group, meaning that the evolutionary history of dicot plants cannot be traced to a single most common recent ancestor. Instead, a number of lineage they diverge earlier than the monocots did. Okay, there's also a 6th way to distinguish monocots from dicots. So the first one is flower. Flower usually arrange their parts in circle with the reproductive parts in the middle surrounded by petals and sepals. In dicots, these flower parts are pentamerous. In other words, the flower part of dicots are arranged, structure, or number in multiple of 5 or sometimes 4. This is not reliable. However, it's not the easiest characteristics to look for in flowers that have their reduced or numerous parts. The second one is leaf venation. Venation refers to the pattern of veins in a leaf blade. These veins are responsible for the transport of water and carbohydrate throughout the plants. In dicot, these veins are arranged in a net-like or reticulated pattern. This vein in such leaf appears to look like a finely branched network throughout the leaf blade, with fine veins reticulating between the major veins. However, as the number of flower parts, leaf venation is also an unreliable characteristic upon which to base your classification assessment. Sunflower may display reticulated venation but are actually monocots, such as the Aroids and Dioscoriales families. The third one is roots. In plants, the radical or embryonic root is the first part to emerge from the seed. It shoots down into the ground and begins taking up nutrients and water from the soil. The radicle of a dicot plant develops into the root of the plant. More specifically, the root of dicot is known as a taproot. The taproot system have a long and deep primary root, with smaller secondary root growth laterally branching off the primary root. So the fourth one is stem. Dicot exhibit secondary growth, which is the ability to increase their diameter via the production of wood and bark. This is the result of two lateral meristem, the cambium and the vascular cambium. This lateral meristem continue to produce new cells throughout the life of the woody dicot plant, ultimately increasing the girth of the plant. The rigidity of wood and bark provide mechanical support against gravity and desiccation to dicots, allowing them to grow large, tall, and solid. In a cross-section of dicot stem, you will find an epidermis, hypodermis, endodermis, ground tissue, and vascular bundles. Typically, dicot stem have the following characteristic: multicellular epidermal hair all over the epidermis, collagematous hypodermis, pith, differentiated ground tissue, and a limited number of vascular bundles in concentric arrangement. The vascular bundles are typically limited to number of 4 or 8 and arranged near the perimeter of the stem in 1 or 2 rings. The fifth one is pollen grain. Pollen grains are like the male sex cells of plant. They are the male gametophytes that produce the plant sperm cells, or male gametes. The pollen grains of dicot plants have physical characteristics that distinguish them from the pollen grains of dicot plants. If you look closely, dicot pollen grains are tricolpate, meaning that they have 3 ridges that go throughout the outer layer. This structure is derived from the first angiosperm, which has monosculated pollen grain. That means having 1 ridge. The monosculated form, however, was not retained over the course of divergent evolution. Okay, the last one is embryo. Embryo is the part of the seeds that contain of all precursor tissue of the plant and one or more cotyledon. As the name suggests, dicots are characterized by having 2 or D cotyledons in the seed and 2 embryonic leaves emerging from the cotyledons. The seed pod of dicot are variable in size, shape, texture, and structure. Dicot seed pod can have almost any number of chambers, including 0. More often than not, dicot seed pods contain more seeds than a monocot seed pod. The cotyledon is the part of the plant to emerge from the seed and is the actual basis for distinguishing 2 main group angiosperms. Cotyledons are important in food absorption and are responsible for absorbing nutrients from the environment until the plant can photosynthesize its own nutrients. Okay, so that's all from me, and thank you.